Race Oncology is a business focused on novel RNA-directed therapeutics, focused on oncology, but equally we're also focused on protecting the heart. I'd like to talk to you about why you should consider investing in Race Oncology. We are a clinical stage oncology company with opportunities in cancer that, when we aggregate and estimate, accrue to over $2.6 billion in opportunity. Importantly, we've got an asset in Xantrine, which is effective. It is the most potent inhibitor against the FTO protein, and that's being independently assessed on a couple of occasions. So that inhibitor is a significant asset with a significant opportunity. When I talk to you today, we've evolved our business. So we're talking about expanded projects, pipeline, and prospects for our business. We've improved the formulation. We're extending the formulation, and that's going to help us to provide additional utility in solid tumor oncology and beyond. We announced recently a cardioprotective preclinical study that demonstrates that we have synergy with doxorubicin and that we also protect the heart, so you get a better cancer outcome and you also get heart protection in the process. That's a significant outcome, and I'll talk to that in more detail. But equally, we've got multiple short and medium-term inflection points ahead of us that we believe will create value for Race and for you as shareholders. So let's talk about our traditional area of focus, which is being oncology. And in oncology, we're currently in a phase 2 trial, in fact, in Israel, in acute myeloid leukemia in a combination trial. But importantly, we're pursuing the extramedullary form of AML, and we've got a trial that's going through ethics in Australia, and which will be in patients in Australia next year. We've also got significant opportunity through Stratum 2 of our EMD Australian trial. That will allow us to evaluate the FTO opportunity. And FTO, of course, for Xantrine is a big, significant opportunity in oncology. Again, we'll come back to that in more detail. I'll talk about cardioprotection. I touched on it earlier, but cardioprotection is a new platform of growth for us. In some respects, it's an easier platform to pursue because the drug can be used as is in its current form without radically changing standard of care practices. So that's a big opportunity now in cardioprotection. And then our strategy equally has evolved to include the opportunity to pursue new RNA-relevant molecules. FTO is a great molecule and a great protein to pursue, but there are others, and we're seeing them, and we'd like to go after them where we think it's relevant and where they can create value. In looking at the market, the cancer market has evolved, of course, over years. So historically, people had a single drug that was applied to multiple indications. It worked for some, it didn't work for others. Regrettably, it could be toxic for some and create adverse effects. What's important for us is that we're going down a road which is precision oncology. And we're able to do that because we've got a drug that we know has efficacy against a particular protein, that being FTO. We know that through biomarkers, we can identify patients that have FTO and where that FTO will synergize positively with Xantrine. So it means that we can target specific patients and have a high degree of confidence around outcomes, and that creates value. We're talking about the mRNA space. So what's important is that when you look at the transition from DNA to RNA and you look at transcription and translation, FTO as a protein is at the center of that. And that's the protein that we've got a significant opportunity against. But what we've also known is that RNA regulation is important biologically to many other diseases, not simply cancer. Equally cardio health, but a whole lot of other areas. But for now, we'll talk about oncology and cardio health as our focused opportunities. FTO is a demethylase regulator, sometimes called an eraser. So when you look at the process of writers and erasers and transcription and translation, you'll find FTO right in the center of it. When you study and read about all the papers written about FTO, it's clear that it's a driver of oncology and of cancers. It's also clear that if you can regulate it and inhibit it in certain cancers, you're having a positive impact on that cancer. We're lucky that we are the most potent inhibitor identified of FTO. And we're lucky that our asset Xantrine is the most clinically advanced inhibitor of FTO. And because we've been in so many patients historically, we have the ability to quickly move into phase 2 trials for Xantrine. But I also don't want to miss that what's different today is that we're clear we will go beyond FTO and Xantrine, and we'll pursue new molecules and new protein targets where we identify them to be of promise. I touched on earlier a number of $2.6 billion, a revenue opportunity for us. If you look broadly at the oncology market, it exceeds $120 billion. It's enormous. What's unique for RACE, for Xantrine, is that if you look at FTO is present in most every cancer. So there is no doubt an opportunity to cross many different cancers with FTO. We're targeting just 3 of those being melanoma, renal cell, clear cell carcinoma, and AML. And when we look at modest share ambitions for those cancers, we think there's a revenue opportunity that exceeds $2.6 billion alone. The other way of looking at what we're worth and what we could be worth in the future is comparative valuations. There is significant deal activity in the RNA space. So companies that are effectively preclinical, they've got assets that are preclinical, haven't been to the clinic, so less developed than ours, are getting significant levels of investment, acquisition, or acquiring themselves. And this slide alone just gives you a view of some of the activity that's happened in the last number of years. It's a very, very attractive space. So let's look at strategy and the choices we choose to make to create value for shareholders and patients in the process. Our 3-pillar strategy is an evolution, but we believe it's a positive evolution because it creates more value for us collectively. We're pursuing through Pillar 1 Xantrine in its current form. So Xantrine today is highly relevant, has been highly developed, and we've got a lot of experience in it. That allows us to go headstrong and quickly into the AML space, which we're doing through our combination trial in Israel, but equally the EMD trial, which will be pivotal in Australia shortly. Equally, we've got a US IND that we plan to launch next year in 2022. And we've got the new cardioprotective insight that we'll touch on in more detail, but which frankly is transformational around our value opportunity. The second pillar we call Xantrine Optimized. It's new formulations and improvements to Xantrine that will make it more relevant to solid tumor oncology. It will make the drug easier to administer, and importantly, it'll help us to drive and develop intellectual property. And finally, beyond Xantrine, new molecules targeting new proteins. We can do that through our own development capabilities that are being enhanced. We can do it through partnerships, and we could do it through acquisitions for that matter. So that's a new opportunity for us. We've got a wealth of opportunities that are either in place, trials that are underway. We're not simply preclinical, we are in clinical trials already. We have additional opportunities in front of them, which this slide addresses, but I'm going to let Daniel in a minute take you through the detail of those. There's some short-term value drivers, of course. We've got our cardioprotective patent and announcement that was recently updated. We've got additional data we hope will flow in that space. We've got EMD, the AML extramedullary program, which is in ethics approval at the moment. That will have a first patient at some point in time in the near future. And we've got our US IND listing that's being pursued. In Pillar 2, we've got our melanoma animal data that follows very significant and strong preclinical results, IV formulations that effectively address Pillar 2 and improvements, and a number of other initiatives that you'll hear about shortly. So I wanted to come back to where I started. Why invest now? Well, we have a clinical asset. Xantrine is not a preclinical asset. It's a clinical asset. It's being in the clinic historically. It's in the clinic right now today. But importantly, it targets indications that are large and which potentially are a significant source of revenue quite quickly. Secondly, against FTO, we have the most potent inhibitor. And we're well advanced relative to our peers. Our drug is more effective than alternatives against FTO, so we're going down a road of developing that opportunity soonest. When you look at what's evolved in strategy, we're no longer a single drug company with a single opportunity. We've got broadened opportunities, we're expanding projects and pipeline. Our new formulations will make us easier and more relevant for administration in different applications in oncology, and the cardioprotective opportunity is unique and large, and we're going quickly down that road. So we have some short-term good news ahead of us. We'll be releasing that as it comes to market appropriately and pursuing those opportunities as quickly as we reasonably can. What I'll do now is hand over to Daniel. Daniel will walk us through a more detailed overview of our respective pillars and programs, and I'm sure you'll find that a significant interest.
Thank you, Phil, for that introduction. So I'd like to take you through a detailed discussion and overview of all the various programs we have underway. As Phil showed, we have a deep pipeline of activities underway, driving value for the company and driving value for shareholders and building what we think is going to be a very valuable company. The first where I will begin is Pillar 1, which is what we consider the current formulation of Zantrine and all the particular clinical and preclinical activities that can make use of that particular formulation. In this particular pillar, we have 4 different programs. The first of these is a combination AML trial that's currently underway, phase 1b/2 trial in Israel. First patient was treated in August of this year. We expect to be able to update the market on progress of that trial sometime in early 2022 as that program progresses. Second program we have well advanced clinical program is in extramedullary AML. I'll talk more about that going forward, but we have 2 what are called stratums. You can think of these kind of almost as independent trials targeting different patient populations in different ways to best make use of the opportunity we have in that particular indication. And the final one we have in there is a phase 2b cardioprotection program and preclinical program making use of the recent discovery that Zantrine can protect the heart while better targeting the cancer. Starting off in relapsed and refractory acute myeloid leukemia. So this is a follow-on phase 1b/2 trial from the phase 2 trial we reported on from last year that used Zantrine as a single agent. That gave quite an attractive, quite an exciting result. About 40% of the patients in that trial responded to the drug. And this is it's quite interesting because these patients were extremely heavily pretreated. They'd failed multiple existing and experimental drugs, and yet they responded to Zantrine on its own. So we were particularly excited by that. So we wanted to follow on from that program and build upon that work, and we decided upon a combination of Zantrine plus 2 nucleoside analogues called fludarabine and clofarabine. This program was built on the basis of work came out of the Baylor College of Medicine, which identified synergy between these 3 drugs that were better able to target AML. This program is being led by Arnon Nagler, who's principal investigator, and his team at the Sheba Medical Center. He was the investigator that ran the first phase 2 trial for us. And we think that this is going to be an exciting use of Zantrine in a more traditional way of using Zantrine. We have decided to target the extramedullary form of AML as our pathway to approval. And the reason why we've chosen this particular form of AML, and I'll explain what this actually means. If you look on the top slide there, you can see these dark patches. This is a PET scan of a patient with extramedullary AML. Extramedullary just means that the AML, or the acute myeloid leukemia, has left the bone marrow where it normally lives and taken up residence somewhere else in the body. And in this particular case, it's taken up in the breast tissue of this particular patient. This is a particularly difficult form of AML to treat. There are no established protocols or licensed treatments for this particular form of AML. This is despite it being relatively common. About 20% of all AML patients happen to have this form. So we think this is a nice niche to go after. It's a very competitive space, acute myeloid leukemia, and you need to have something that you can differentiate from all the other drugs that are on the market or in development. The reason why we chose the extramedullary form of acute myeloid leukemia is in the phase 2 trial that we ran in 2020, All 4 of the 4 responders in that trial all had the extramedullary form of AML. So we think there's something about extramedullary AML that makes it acutely sensitive to Xantrine. So one of the nice side effects of using extramedullary as a form is that it's an ultra-rare orphan indication, meaning that you only need small numbers of patients, relatively low cost, to get all the way through to approval. So we've decided to split this program into 2 stratum, a traditional one where a high dose chemotherapeutic type approach and a low dose FTO directed. So the low dose is the most exciting from my perspective, as it utilises the known ability of Xantrine to inhibit FTO. So it's in combination with a particular drug that's used in AML and a few other leukemias as well, called decitabine. And this is designed to target and kill cancers using very low, non-toxic doses of Xantrine. Unfortunately, Xantrine, when given at high doses, is quite toxic, like all chemotherapeutic drugs. But at low dose, we're able to achieve a result, at least preclinically, that kills the cancer using very, very low doses of the drug. We have a clinical program well advanced to study this particular set of patients. The human ethics was filed in October of 2021, and we expect to have an outcome of that in the next couple of months. The nice advantage of this is it provides proof of concept for the FTO opportunity. So while there's a lot of preclinical data to support FTO, there's no clinical data yet, or no direct clinical data, and this trial will most quickly generate data that will support the FTO concept. Also within this pillar is what we consider what we're calling the anthracycline cardioprotection. So anthracyclines are a class of chemotherapeutic agents, the most widely used, the most effective drug, been around for 60 odd years, but they come with a really serious side effect, which is permanent damage to the heart. This is such a problem that many oncologists are reluctant to use anthracyclines or limit the dosing of anthracyclines that they use in a patient, and that can compromise the treatment of the patient. The other thing that can happen is that patients, as they're now living much longer with cancer, are being cured of their cancer, but going on to suffer long-term consequences of cardio damage and ultimately dying of cardiac disease, which is not a great outcome for the patient. So it's been known for a long time that Xantrine had a lower cardiotoxicity than other drugs in the class, but it wasn't known how this particular mechanism of action worked. So we designed a program at RACE to investigate this, and we wanted to find out, could Xantrine protect the heart from anthracycline-induced cardiotoxicity? And as we reported very recently, it actually can. And this is quite I'm going to say amazing discovery, because it also is able to better target the cancer. And the end result of this is we get a protection from the worst side effect of chemotherapy, the part that you don't recover from, and whilst better targeting the cancer. And so it's kind of the best of both worlds. One of the great advantages of this particular market is it's very large and it's an existing market. So millions of patients are treated every year with anthracyclines. And if we can make those anthracyclines not only better at targeting the cancer, but less dangerous to the patient, we think we have something that'll be very attractive to both clinicians, patients, and large pharma. And so how do we go about tackling this? Well, we've got preclinical programs underway, and we'll continue that work, particularly in animal and further in cell cultures. But we intend to, or in advanced discussion with running a phase 2B clinical trial in breast cancer patients who are at high risk of cardio damage from anthracyclines. So about a third of all breast cancer patients are treated with an anthracycline at this point, and many of those patients suffer long-term heart damage consequences for that. Or for those patients who already have existing cardiac disease, which is obviously very common in the patient population that tends to get breast cancer, they can't be treated at all currently, or can be treated only with very low doses. And so the expectation here is this trial will be able to improve the outcome for these patients, provide them with much more effective treatment without that risk of cardiac damage. We can target this huge market with a high unmet need. And we also have the potential there, this is something that will be explored preclinically and ultimately clinically if everything pans out, is the use of Xantrine beyond just within the oncology market. Heart failure and heart disease is something that exists beyond cancer. Very exciting opportunity. You can see here, just before the slide changes, you can see here that as you have increasing concentrations of Xantrine, you get increasing viability, rescuing of the cells from dying, and these are heart cardiomyocytes, which are muscle cells. Really quite an amazing result there. So we move to what we call the new pillar 2, which is Xantrine that's been optimized for targeting FTO. This is quite a large program. I won't go through all of these different areas here in detail, and I'll look at them individually. But the major problem with targeting solid tumours or other indications is that the current formulation of Xantrine has to be delivered by a central line infusion, so into the chest, directly into the aorta or the heart, and over a 2-hour infusion. This is very disruptive for the patient. It's a risky procedure for the patient, and it requires often that they remain in hospital and can't be treated as an outpatient type basis. So what we want to do is to be able to make a form of an approach with Xantrine, a new formulation of Xantrine that allows this to be used peripherally in a whole range of different types of cancers. So you obviously want to be able to look at cancers that require FTO to survive, so what we're considering FTO-addicted cancers, and the clear cell renal cell carcinoma is a good example of that. You also want to be able to use it in combination with other drugs, so it's been discovered that FTO inhibition shows synergy with a whole range of other combination drugs, and in order to do that you need to have a formulation that works well with those particular combinations. And you also can overcome resistance to these other drugs that are used in existence. So we've decided to undertake a phase 1/2 combined solid tumour clinical trials enriched for the types of cancers that we expect to respond. So in particular case, melanoma and clear cell renal combination, clear cell renal cell carcinoma, and various drug combinations that we think are most likely to work through our preclinical activity. The advantage of this is we get to sample across a wide range of different tumour types and find in the patients where this is most likely to work. We have an ongoing preclinical program exploring the use of Xantrine in melanoma. We just reported recently in the last month or so interesting preclinical results where we found that melanomas that overexpress FTO are exquisitely sensitive to Xantrine, and also that opens up the opportunity through other published work that has been done by other groups that inhibiting FTO using a drug like Xantrine can enhance and overcome checkpoint inhibitor or immunotherapy resistance, which is a major problem. So melanoma is one of those cancers where immune therapy has made a major impact. 10 years ago, if you got advanced melanoma, your expectations of a long life were not good. And then 10 years ago, the immunotherapy drugs came onto the market, the Keytrudas, and they revolutionized the treatment of advanced melanoma. And patients went from being weeks from dying being so advanced to being completely cured. And that's something that amazed and shocked doctors and everyone. And these class of drugs have gone on to become the largest oncology market class of drugs, about $70 billion. The downside is they only work in melanoma a little under half the time. And whilst if you're lucky and the immune therapy drugs work for you, it's great. If you're unlucky, then there's not much the doctors can do for you. And at this point, we think we may have an opportunity here to make those tumours that don't respond to the checkpoint inhibitors, make them respond through inhibiting FTO. And there's a lot of preclinical data to support that hypothesis. So we have a program underway, preclinical, which we're reporting on, and the intention is we'll have a proof of concept clinical trial to start as soon as we have the animal testing. Offers a huge amount of potential value and opportunity as a proof of concept, particularly of the concept of utilising FTO inhibition as a way of enhancing immunotherapy. And this goes across a lot of different cancer types. Immunotherapy drugs are used in a wide range of different cancers, including breast and lung and gastrointestinal. The other cancer we're targeting as our sort of proof of concept is clear cell renal carcinoma. And the reason why we've chosen this one is there is good preclinical evidence that FTO is essential for this particular cancer, or for the majority of cancers that form in this particular one. About 90% of all clear cell renal cell carcinomas have a particular mutation, driver mutation, in this gene called the von Hippel-Lindau gene, or VHL gene. And when that gets mutations or gets switched off through various mechanisms, then the cancer becomes very sensitive to inhibition of FTO. And so we have a preclinical program underway, studying this to see whether xantrine can work in clear cell renal cell carcinoma, and we're expecting to be able to report news on this progress of this trial shortly. And obviously the next stage with this, if it all works out really well in the preclinical, is to move it into animal models and then into the clinic rapidly. And we think these programs are all complementary with one another, and we can make use of what we know to enrich in some of these generalised trials to attract patients with these particular types of cancers into them. Obviously, there's significant commercial opportunity here. Clear cell renal cell carcinoma is a serious cancer with a low survival rate and a real need for better treatment options. Obviously, underpinning all of this, there's no point saying we have a precision oncology drug if we don't have some means of identifying which patients will respond to xantrine. And so the modern approach to anti-cancer drug development is to develop companion diagnostics, which both diagnose and allow you to monitor the treatment whilst the patient is undergoing treatment with your drug. So in order to do that, you need to identify what's known as a biomarker, something that correlates with response in a patient. So you want to be able to say, this particular gene's present, this protein's present, these series of, or maybe a series of proteins are present. And the end result of that is that you end up with a marker which you can say, you have a patient, you can screen them before treatment, and you can say, you're very likely to respond to xantrine, or you're very unlikely to respond to xantrine, and you only give the drug to the patients that will respond. So this is such an important area of activity for us. We have two programs underway, one with Professor Damasi at the Chimes Sheba Hospital, another one at the University of Newcastle with Professor Murray Cairns, tackling this question in two different ways. One of it kind of a top-down global system, and then another one from a bottom-up genomics-based, looking at individual transcripts and the methylation status of individual transcripts. Both are very complementary with one another. They both help one another and allow us to develop gather the data required to create a companion diagnostic. We expect to be able to report news of this program over 2022. We think this will add a great deal of value to Xantrine and to the company. I've been talking about formulation program, about needing to improve the delivery of Xantrine. The problem with Xantrine, as I mentioned before, is it requires this 2-hour central line infusion. It makes that complicated and difficult to deliver in many oncology indications and many the way the oncologists like to work. They like to be able to deliver a drug maybe in an outpatient, maybe in as an oral pill that the patient can take at home. They want to try and make it as easy for the patient and as least disrupting for the patient as possible. So we've developed and been working on this program for quite some time. We have established a relationship with the University of Wollongong to start to screen some of the various formulations we've been working on. And we think that we have various opportunities, a number of them, that will allow us to develop a form of Xantrine that can be delivered through a peripheral vein in an arm or a leg that's much easier to utilise in an outpatient environment or in the patient's home. The other thing this provides is IP protection around the drug. It effectively resets the patent clock and gives us protection for another 20 years going forward. So adds a huge amount. Not only does it expand the potential market for the drug into different indications, but it also gives us longer protection. This brings us to Pillar 3, the new Pillar 3, which is we've titled Beyond Xantrine. We've developed a lot of skills and knowledge in this new space of M6A RNA methylation regulation, and it's a really exciting area. It's a cutting edge area. There's a lot of companies out there pursuing this, but it's a relatively new area, and there is a relatively small number of companies that have the skill sets required to actually exploit this opportunity. So we think we can build on what we've actually done and learnt through Xantrine. We have the skills in-house to be able to actually start to develop new molecules that can target not only FTO in different ways, and this can become really important. Not every indication Xantrine will be most important to do. So it's always best to have more than one molecule targeting a particular pathway. The other thing there is this particular pathway, the M6A RNA pathway, has multiple proteins in it. They all do different things, and sometimes it's not FTO that gets dysregulated, it's another protein, for example, metal 3 or metal 16 or metal 14 or ALK-BH5. These are all proteins that are in this pathway, and you would like to have drugs that target each one of those different proteins so that you can actually match with a companion diagnostic, you can match the treatment to whatever the particular mutation this happens to do. So this is a really exciting area, not only within oncology, but beyond oncology, builds and de-risks the company from the perspective of being solely focused on a single drug, but it also provides new IP and new opportunities for the company, making use of the skill sets that we have. It's a relatively cheap program for us to undertake at this point in time, under $1 million. So we think that this can actually generate far more than $1 million worth of value.
Yeah, thanks Daniel for that program overview. Clearly we've got a wealth of opportunities and we're progressing them rapidly, but it's a bigger opportunity than we've ever had, so we're pleased to have that in front of us. Just taking you through the corporate overview, we've got a very experienced board of directors, Dr. John Cullity, our chair, who's got a deal background, a pharma background. He's New York based, and when appropriate, he's the right person to help us, frankly, market our company to potential investors, partners and other. Daniel, you've heard from today. Daniel's being a big driver of much of strategy over the last couple of years. Daniel's got a genomics background and runs his own genomics business, Nucleic. He's very active in leading our science part of our business and working with myself. My background is with Johnson & Johnson in consumer marketing and consumer healthcare. We've got an independent director in Mary Harney. So Mary's got a great governance background, comes out of Melbourne, has worked extensively with academic institutions, and Mary's an effective contributor to the board. I'll highlight on management, Dr. David Fuller joined us about 6 months ago now. David's got a big oncology background. He worked leading a CRO on the oncology practice across Asia. So David leads as our Chief Medical Officer and does a good job in that regard, ably supported by Marinella Messina, who's been with us for a longer period of time. So Marinella is very active helping us on our programs. I will also mention Dr. Mike Kelso. Mike joined us recently from the University of Wollongong. You may be aware that we've now got a program in place with Wollongong, and Mike's got great preclinical and drug development expertise, very much broadening our capability, including an associate of his, Dr. Ben Buckley, that's recently joined. So great capability in that space. We're often asked about our IP portfolio. We would use the language to say our portfolio has grown. We've now got 6 US patents. We talk about our patent thicket. It's getting thicker and, if you like, harder to penetrate. So we're building IP. As always, when people ask, well, how do I value the IP? Well, frankly, you have to value it in the eyes of how other people view it. But we believe we've got a great IP package that protects us, our asset, and allows us to market our asset accordingly. If you look at our corporate snapshot, our market cap is at about, $560 million, share price of $377. We've had, as you can see from the share chart over the last 12 months, a significant run up as the market has appreciated the value that exists in the FTO opportunity. And we think ahead of us that the value that the cardioprotective opportunity creates will equally be larger. But we've got much runway ahead, but we need to progress our plans. We need to invest to be able to execute those plans and deliver the results that will make a difference. I should also mention on our significant shareholders, Daniel, our CSO. Daniel's now at 9%, Dr. John Cullity at 6%. So the management team are large holders of our company, and Merchant Opportunities Fund out of Perth, a long-term supporter of Race Oncology. So you've heard today that we've got a great rich history with Xantrine in the clinic, and because of that rich history, we're able to go very quickly after the FTO opportunity. And you've seen the preclinical results. You're aware of our clinical plans against FTO. They'll go very quickly through the EMD Stratum 2 opportunity that Daniel touched on. But equally next year in 2022, we'll go after a phase 2 trial that we hope to take out against a number of different solid tumor oncology opportunities that will demonstrate its utility more broadly. So FTO is important and will be important, and we're going to maximize its value. But equally, I would say in cardioprotection, we've got a whole new leg and opportunity of growth. We've got great preclinical results. We hope to generate more, but importantly, we'll take that quickly into the clinic through a proposed phase 2 trial. With our drug development capability, formulation improvements, I think, if you like, those broad opportunities mean that we're better placed than we ever have been for creating, frankly, shareholder growth, but importantly, value for patients. Ultimately, that's what it's about, and that's what we hope to create going forward. Thank you.